All right, welcome biologists to our little podcast about experimental design. You all have done experimental design, knowing how to set up an experiment since you were very young, little tiny people, probably in 5th grade, 6th grade. And so hopefully this will be a review for everyone. Whether it's a review or a first time for you, then you just please let me know if you have any questions, okay? This will be pretty important for us as we proceed through the year. So let's say you were doing some stuff and you observed something, and after observing some problem or whatever, some phenomenon that raised a question for you, and after doing some background research and developing a hypothesis, and after you determined your hypothesis, you realize you need to test that hypothesis, okay? You need to test it to see if it's on the right track. There are several ways you can test a hypothesis. Depending on what your subject of interest is, what your question is, what your hypothesis is, the best way to answer that hypothesis might be simply to do observations of something. If you want to know what cardinal behavior is in the wild, you might go and observe little cardinal birds and see how their behavior and document what you see. That's a field study. But another type of way we can test a hypothesis, if our hypothesis is trying to see if one thing affects something else, then we'd want to set up a controlled experiment. Maybe we want to know if color of light affects plant growth, for example. Then that is a subject where we would set up an experiment in a controlled situation in a lab and determine the result. Okay? So, a controlled experiment then is designed to test a hypothesis, and the elements of this experiment are at least 2 setups of the experiment. For example, if I'm doing I want to test if different colors of light have an effect on plant growth, then I would need at least 2 setups. I would need a control setup and some experimental setup, okay? So I would need an experimental treatment, which is a setup of the experiment that is going to have something weird done to it. For example, maybe I want to see if, red light or blue light or purple light or green light or whatever has an effect on plant growth. Maybe I want to test all those colors. So I would have to have a setup of my experiment, which would mean a plant and a light and a spot and all those things for each color of light that I was going to test, okay? But there would also have to be, and this is really important, being a, let me get my little pen up here. There we go. This is really important, being that this is a controlled experiment we're talking about. We need we need one setup of the experiment that is going to have absolutely nothing weird done to it. Perhaps we need a plant that is just grown in normal lighting or something like that. But it's not going to have anything unusual done to it, okay? This control treatment serves as our standard of comparison. It is going to be the way that we know we've got, okay, so this is what normal looks like, and if we get something that grows like this and something that grows like that, whereas normal grew like this, we can tell what kind of effect our experimental treatments are having on our plants, okay? So the control treatment is really important because it allows you to compare a normal situation to the experimental situation so that you can determine if your experimental variable is having an effect on your results, okay? Now, remember the term variable. Variables are anything that is altered or that varies among these setups, the various treatments, the normal light, the blue light, the green light, the red light of your experiment. Now, in an ideal, perfect world, nothing would vary between the different setups except the one thing you are trying to test, the color of light. Okay, that would be the only thing. Otherwise, all the plants are identical, all the pots they're in, all the soil they're in, all the temperatures, everything's identical. The only thing that varies is the color of light. Why? Because if you have more than one variable, maybe you've changed the soil type from one plant to another. How do you know if it's not the soil then that's affecting your results? You have to be able to know exactly that the light is the one thing that is affecting any differences in growth that you see. So that's really important. The control treatment is super, super important. Okay? So, constants. Constants. Constants are everything that is kept the same between those treatments. So constants are absolutely critical. If we vary the soil, then the soil is no longer constant. It's another variable, and it could affect our results. So constants are absolutely crucial. You have to keep as many things constant as possible among your different experimental treatments. Okay? Now, among our things that are going to vary, there are two particularly important variables. One is the independent variable, and the other is the dependent variable. Okay? The independent variable is the variable that is purposefully altered by you in the experimental treatment. So in our imaginary experiment, the color of light is the independent variable. It is the one thing that you are changing from this plant to this plant to this plant to this plant. You've got the green light, the red light, the blue light, and the normal light. Okay? So those different colors might be called levels of your independent variable. The independent variable is light type. Okay? The different levels would be red and blue and green and whatever other colors you had picked. Okay, oh, there we go. I've already written this down. Okay, so those are your different levels. Now the dependent variable is the thing that is measured, that you are going to measure as your experimental results. So this is what you're measuring. So in this experiment, maybe we had decided we were going to measure the height of the plant. Does color of light affect the height of the plant? Okay? Now, trials. Trials are really important in a controlled experiment. Having lots of them is really important. A trial is nothing more than a repetition of an experiment. Okay? We want to do as many trials as possible. Now, in our class, we are fairly limited as to how many trials we can do. Usually, a trial, we're going to consider each lab group a trial. We certainly want to have at least 3 trials in any experiment, okay? 3 repetitions, okay? Trials exist not because if we do a gazillion trials, we will eliminate errors, we will eliminate mistakes in our experiment. That is certainly not true. We will not eliminate mistakes. We will never do that. I wish we could. But what trials, multiple trials, do is they reduce the effects of errors on your results. If you have just one grade in my class for 9 weeks and it was one test and you were sick that day and you did not do well and you got a 50, okay, and that's your 9 weeks grade, you are going to really wish we had had multiple trials for your grade. You are going to wish we had had more grades to fill in, because then that would have reduced the effects of that one bad day on your grade as a whole. Okay? If we run our experiment, green light, blue light, red light, clear light, we run it, we do it once, we get results. We do the whole thing again, we do the whole thing again, we do the whole thing again, maybe 5, 10 times. If we had one run where the plants just all died because you forgot to water them for a week, okay? If that was your only trial, all your plants are dead, you have no data. But if that's one trial out of 10, then we can reduce the effects of that one bad trial because we have 9 others to look at, okay? So they trials reduce the effects of error on your results. Okay, so those are just some basic things to remember about experimental design, and we will be hopefully designing an experiment in class next time I see you. So hope you have a great evening, and until next time, take care.